We will take the example of insect resistant genetically modified crops. So, in the recent years, several crops, food crops as well as commercial crops, that are seriously affected by insect pests have been subjected to recombinant DNA technology to control the insect pests, and insect resistance has been achieved in such plants. Many insects like bollworms, beetles, affect the crops. However, these insects are known to be destroyed by a bacterium if it infects these insects. So, there is a bacterium belonging to the species Bacillus thuringiensis. This bacterium can parasitize the insect pests, and wherever the insect pests are found to be infected with Bacillus thuringiensis, the pests are known to die. The reason for this is that Bacillus thuringiensis produces a protein toxin, which is stored in the crystal form in the cysts of the bacteria. The nature of this toxin varies. The main toxin, which is primarily a protein because it is in the crystal form, the main toxin is represented as cry because of its crystal form as cry. There are variants. Like, for example, cry 1, cry 2, and so on, for different groups of insects on which these bacteria infect. Cry 1 has further variants. Cry 1A, Cry 1B. Similarly, in Cry 2 also there are variants like Cry 2A, Cry 2B, Cry 2C, and so on, depending on the insect host. Further variation may be found within Cry 1A when it is designated as, for example, Cry 1A may have Cry 1AA, Cry 1AB, Cry 1AC, and so on. Each of them is specific to a particular species of insect. The cotton plants all over the world are infected by a group of insects called bollworms. A group of insects called bollworms. These bollworms can be destroyed by the toxin Cry 1 AC and Cry 2 AB. Each of these have slightly different gene sequences. So all these gene sequences have been identified, and the various genes have been artificially synthesized for the production of these toxins, and those genes are now being introduced into the plant varieties to control the pests. For example, the cotton plant infected by bollworms, to control bollworms, the Cry 1AC and Cry 2AB genes have been introduced. As a result, a new variety of genetically modified cotton plant has been obtained, and this cotton plant which is resistant to the bollworms is called BT cotton. So this BT cotton, in case it is infected by the bollworms, because of the production of toxins in the cotton fibers by the newly introduced genes, the bollworms get destroyed and the yield is normal.